Dear viewers, this is part 1 of the video series on the gene control of cell function. This figure 1.1 shows the gene control of cell function. From DNA, RNAs are formed. RNA comes outside the nucleus and forms various proteins for various functions and cell structures. Genetic control of cell function. See figure already shown. 1.1 Control of cell function. Genes, which are located in the nuclei of all cells of the body, control heredity from parents to children, as well as the daily functioning of all the body cells. The genes control cell function by determining which structures, enzymes, and chemicals are synthesized within the cell. See the figure 1.1 again. It shows the general schema of genetic control. Each gene, which is composed of deoxyribonucleic acid, that is DNA, controls the formation of another nucleic acid, ribonucleic acid, RNA. This RNA then spreads throughout the cell to control formation of a specific protein. The entire process from transcription, that is copying of the genetic code in the nucleus, to translation of the RNA code and the formation of proteins in the cell cytoplasm is often referred to as gene expression. Because the human body has approximately 20 to 25,000 different genes that code for proteins in each cell, it is possible to form a large number of different cellular proteins. In fact, RNA molecules transcribed from the same segment of DNA, the same gene, can be processed in more than one way by the cell, giving rise to alternate versions of the protein. The total number of different proteins produced by the various cell types in humans is estimated to be at least 100,000. Some of the cellular proteins are structural proteins, which in association with various lipids and carbohydrates form structures of the various intracellular organelles. However, most of the proteins are enzymes that catalyze different chemical reactions in the cells. For example, enzymes promote all the oxidative reactions that supply energy to the cell, along with synthesis of all the cell chemicals such as lipids, glycogen, and adenosine triphosphate, that is ATP. Dear listeners, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Figure 1.2 shows the helical double-stranded structure of the gene. The outside of the helix are strands composed of phosphoric acid and sugar deoxyribose, that is sugar phosphate backbone. The inside between the two coils are the base pairs of DNA double helix. Recall the figure 1.2 DNA structure. Cell nucleus genes control protein synthesis. In the cell nucleus, large numbers of genes are attached end on end in extremely long double-stranded helical molecules of DNA having molecular weights measured in the billions. A very short segment of such a molecule is shown in figure 1.2. This molecule is composed of several simple chemical compounds bound together in a regular pattern. Figure 1.3 shows the basic building block of DNA. There are a sugar and phosphoric acid molecule which is attached to one of the purine or pyrimidine bases, which is again attached in the reverse manner with a pyrimidine or purine base, a sugar and a phosphate. Building blocks of DNA. See figure 1.3, building blocks of DNA shown earlier. Figure shows the basic chemical compounds involved in the formation of DNA. These compounds include the following: 1. Phosphoric acid, 2. A sugar called deoxyribose, and 3. 4 nitrogenous bases, 2 purines, adenine and guanine, and 2 pyrimidines, thymine and cytosine. The phosphoric acid and deoxyribose form the two helical strands that are the backbone of the DNA molecule, and the nitrogenous bases lie between the two strands and connect them. Nucleotides. The first stage of DNA formation is to combine one molecule of phosphoric acid, one molecule of deoxyribose, and one of the 4 bases to form an acidic nucleotide. 4 separate nucleotides are thus formed, one for each of the 4 bases: deoxyadenylic, deoxythymidyllic, deoxyguanylyl, and deoxycytidylic acids. Nucleotides are organized to form 2 strands of DNA loosely bound to each other. Figure 1.2 earlier shown, it shows the manner in which multiple nucleotides are bound together to form 2 strands of DNA. The 2 strands are in turn loosely bonded with each other by weak cross linkages. Note that the backbone of each DNA strand is composed of alternating phosphoric acid and deoxyribose molecules. In turn, purine and pyrimidine bases are attached to the sides of the deoxyribose molecules. Then, by means of loose hydrogen bonds between the purine and pyrimidine bases, the 2 respective DNA strands are held together. Note the following caveats, however. 1. Each purine base, adenine, of one strand always bonds with a pyrimidine base, thymine, of the other strand, A and T or T and A. Each purine base, guanine, always bonds with a pyrimidine base, cytosine, G with C or C with G. Because of the looseness of the hydrogen bonds, the 2 strands can pull apart with ease. And they do this so many times during the course of their function in the cell. 10 pairs of nucleotides are present in each full turn of the helix in the DNA molecule. Genetic code. The importance of DNA lies in its ability to control the formation of proteins in the cell, which it achieves by means of a genetic code. That is, when the 2 strands of a DNA molecule are split apart, the purine and pyrimidine bases projecting to the sides of each DNA strand are exposed. It is these projecting bases that form the genetic code. The genetic code consists of successive triplets of bases. That is, each of 3 successive bases is a code word. The successive triplets eventually control the sequence of amino acids in a protein molecule that is to be synthesized in the cell. For example, the sequence of bases reading from left to right, the genetic code GGC sequence means the amino acid proline. AGA sequence means the amino acid serine. CTT sequence means the amino acid glutamic acid in a newly formed molecule of protein. Transcription, that is, transfer of cell nucleus DNA code to cytoplasm RNA code. Because DNA is located in the cell nucleus, yet most of the cell functions are carried out in the cytoplasm. There must be some means for DNA genes of the nucleus to control chemical reactions of the cytoplasm. This control is achieved through the intermediary of another type of nucleic acid, RNA, the formation of which is controlled by DNA of the nucleus. The code is transferred to RNA in a process called transcription. The RNA in turn diffuses from the nucleus through the nuclear pores into the cytoplasmic compartment where it controls protein synthesis. RNA is synthesized in the nucleus from a DNA template. During RNA synthesis, the two strands of DNA separate temporarily. One of these strands is used as a template for synthesis of an RNA molecule. The code triplets in the DNA result in the formation of complementary code triplets called codons in the RNA. These codons in turn will control the sequence of amino acids in a protein to be synthesized in the cell cytoplasm. Building blocks of RNA. The basic building blocks of RNA are almost the same as those of DNA except for two differences. First, the sugar deoxyribose is not used in RNA formation. In its place is another sugar of slightly different composition, ribose. Second, thymine is replaced by another pyrimidine, uracil. Formation of RNA nucleotides. The basic building blocks of RNA form RNA nucleotides exactly as described previously for DNA synthesis. Here again, 4 separate nucleotides are used to form RNA. These nucleotides contain the bases adenine, guanine, cytosine, and uracil. Note that these bases are the same as in DNA, except that uracil in RNA replaces thymine in DNA. Activation of RNA nucleotides. The next step in the synthesis of RNA is activation of RNA nucleotides by an enzyme, RNA polymerase. The result of this activation process is that large quantities of ATP energy are made available to each of the nucleotides. This energy is used to promote chemical reactions that add each new RNA nucleotide at the end of the developing RNA chain. RNA chain assembly from activated nucleotides using the DNA strand as a template. Assembly of RNA is accomplished under the influence of an enzyme, RNA polymerase. This large protein enzyme has many functional properties necessary for formation of RNA as follows. 1. In the DNA strand immediately ahead of the gene to be transcribed is a sequence of nucleotides called the promoter. The RNA polymerase has an appropriate complementary structure that recognizes this promoter and becomes attached to it, which is the essential step for initiating the formation of RNA. 2. After the RNA polymerase attaches to the promoter, the polymerase causes unwinding of about 2 turns of the DNA helix and separation of the unwound portions of the 2 strands. 3. The polymerase then moves along the DNA strand, temporarily unwinding and separating the 2 DNA strands at each stage of its movement. As it moves along, at each stage, it adds a new activated RNA nucleotide to the end of the newly forming RNA chain. When the RNA polymerase reaches the end of the DNA gene, it encounters a new sequence of DNA nucleotides called the chain terminating sequence, which causes the polymerase and the newly formed RNA chain to break away from the DNA strand. The polymerase then can be used again and again to form more new chains. Therefore, The code that is present in the DNA strand is eventually transmitted in complementary form to the RNA chain. The ribose nucleotide bases always combines with the deoxyribose bases in the following combinations: DNA base and the RNA base, guanine with cytosine, cytosine with guanine, adenine with uracil, thymine with adenine. There are several different types of RNA. As research on RNA has continued to advance, many different types of RNA have been discovered. Some types of RNA are involved in protein synthesis, whereas other types serve gene regulatory functions or are involved in post-transcriptional modifications of RNA. The functions of some types of RNA, especially those that do not appear to code for proteins, are still mysterious. The following 6 types of RNA play independent and different roles in protein synthesis. 1. Precursor messenger RNA, pre-mRNA, is a large immature single strand of RNA that is processed in the nucleus to form mature messenger RNA, mRNA. The pre-RNA includes 2 different types of segments called introns, which are removed by a process called splicing, and exons, which are retained in the final mRNA. 2. Small nuclear RNA, sRNA, directs the splicing of pre-mRNA to form mRNA. 3. Messenger RNA, mRNA, carries the genetic code to the cytoplasm for controlling the type of protein formed. 4. Transfer RNA, tRNA, transports activated amino acids to the ribosomes to be used in assembling the protein molecule. 5. Ribosomal RNA, along with about 75 different proteins, forms ribosomes, the physical and chemical structures on which protein molecules are actually assembled. 6. MicroRNAs, miRNAs, are single-stranded RNA molecules of 21 to 23 nucleotides that can regulate gene transcription and translation. Messenger RNA, the codons. Messenger RNA molecules are long single RNA strands that are suspended in the cytoplasm. These molecules are composed of several hundred to several thousand RNA nucleotides in unpaired strands, and they contain codons that are exactly complementary to the code triplets of the DNA genes. There are RNA codons for the 20 common amino acids found in protein molecules. Note that most of the amino acids are represented by more than one codon. Also, one codon represents the signal start manufacturing the protein molecule. AUG is the codon, and 3 codons represent stop manufacturing the protein molecule, coded by UAA, UAG, and UGA. The figure 1.4 shows the assembly parts of protein formation. Shown are the mRNA, a tape-like structure, ribosome, transfer RNA, and the protein assembly by amino acids, one amino acid at a time. Transfer RNA, the anticodons. See figure 1.4 shown earlier, that is formation of protein diagram. Another type of RNA that is essential for protein synthesis is called transfer RNA, tRNA, because it transfers amino acids to protein molecules as the protein is being synthesized. Each type of tRNA combines specifically with one of the 20 amino acids that are to be incorporated into proteins. The tRNA then acts as a carrier to transport its specific type of amino acid to the ribosomes, where protein molecules are forming. In the ribosomes, each specific type of tRNA recognizes a particular codon on the mRNA described later and thereby delivers the appropriate amino acid to the appropriate place in the chain of the newly forming protein molecule. Transfer RNA, which contains only about 80 nucleotides, is a relatively small molecule in comparison with mRNA. It is a folded chain of nucleotides with a clover leaf appearance, similar to that shown in figure 1.4. Because the function of tRNA is to cause attachment of a specific amino acid to a forming protein chain, it is essential that each type of tRNA also have specificity for a particular codon in the mRNA. The specific code in the tRNA that allows it to recognize a specific codon is again a triplet of nucleotide bases and is called an anticodon. This anticodon is located approximately in the middle of the tRNA molecule at the bottom of the cloverleaf configuration shown in figure 1.4. During formation of the protein molecule, the anticodon bases combine loosely by hydrogen bonding with the codon bases of the mRNA. In this way, the respective amino acids are lined up one after another along the mRNA chain, thus establishing the appropriate sequence of amino acids in the newly forming protein molecule. Dear viewers, this is the end of part 1 in the 4-part series on genetic control of cell function in the Medicine for Common Man series. Thank you for watching.